Today we're making elephant toothpaste. It's called this because it looks like toothpaste that's so large that an elephant could use it. But don't try putting this one in your mouth. To do this, we need 3% hydrogen peroxide, like the kind you can get at the drugstore, dry yeast, and dish soap. Okay, so the first thing that we're going to do is we're going to take a tablespoon of dry yeast, and we're going to mix that with about 3 tablespoons of warm water. You want it warm but not too hot. If it's too hot, it will kill the yeast. Stir that up a bit. Next, I'm going to take about 100 milliliters, or about half a cup, of 3% hydrogen peroxide. I'm going to pour that into my bottle. I'm also going to take some dish soap. I'm going to squirt that in my bottle as well. Gently mix that up. I'm going to add some food coloring, which is not necessary, but makes it fun. Now, I'm going to add my yeast solution to my baking soda, to my peroxide solution. And there you have it. I have seen some instructions online that call for using 30% or higher concentration of hydrogen peroxide. This is unnecessary and unsafe. I've also seen directions on how to concentrate your own hydrogen peroxide up to 30% from the 3% kind that you can find at drugstores. Again, this is unnecessary and unsafe. The principle behind this is that water boils at about 100 degrees Celsius and hydrogen peroxide boils at about 150 degrees Celsius. So the idea is that you boil it just hot enough that the water will evaporate, but the peroxide will not. The problem is that this is very difficult to control. Hydrogen peroxide vapor is not safe to breathe, and it also produces oxygen gas and heat, which is an ignition risk. The heat that this reaction gives off makes it additionally difficult to control the temperature. So I repeat, do not try to concentrate your own hydrogen peroxide in your kitchen. It's not necessary for this experiment. However, the reaction that we'll be demonstrating is basically the same reaction, but in a safer and cooler looking way. Hydrogen peroxide is an unstable molecule. It will decompose in the presence of light, heat, and just time. That's why the hydrogen peroxide that you buy at the drugstore comes in those opaque brown bottles, That's to keep the light out, to slow the rate of decomposition. Under the right conditions, the hydrogen peroxide will be fine to store in your cupboard for a while, and it will not decompose significantly. But given enough time, it eventually will. We can speed up this process by using light heat, or something called a catalyst. In this demonstration, we're going to be showing how a catalyst speeds this reaction up. Here is the basic reaction. You can see that 2 molecules of hydrogen peroxide, H2O2, will decompose into 2 molecules of water, H2O, and 1 molecule of oxygen gas, O2. Under normal conditions, this reaction is not happening fast enough to see. But when we add a catalyst, all of a sudden, this decomposition is happening very quickly. You can see the oxygen gas in the form of bubbles, and the dish soap traps those bubbles, making it appear foamy. Because we've sped this reaction up using a catalyst, we get to see all of these bubbles being produced at once, and that's what gives us this impressive display. So what exactly is a catalyst? In simple terms, a catalyst speeds up a reaction. However, this isn't the complete answer. Think about it this way. There are 8 billion people living on this planet. The odds that someone living in China and someone living in France will bump into each other randomly on June 24th at exactly noon are very low. It's not impossible. People move around all the time. But it's very unlikely. Chemical reactions work the same way. In order for them to happen, they have to come into contact with each other, and they have to happen under the right conditions. A catalyst is kind of like a matchmaker. In biology, most of the catalysts that we talk about are enzymes, and most enzymes work by bringing 2 or more pieces of a reaction together and holding them in place long enough for them to interact. They can also act as a surface for electrons or atoms to move around, or provide electrostatic forces that help make bonds easier to break. This is kind of like introducing your friend from China to your friend from France. Catalysts aren't part of the reaction, and they're not used up by the reaction. What catalysts do is lower the activation energy, which is the amount of energy required for a reaction to proceed. This is like lowering the odds that 2 people will meet. And there are 2 common ways that this demonstration is done. The first is using potassium iodide. The iodide ion pulls on the oxygen in the hydrogen peroxide, forming hypoiodite. Then it reacts with another molecule of hydrogen peroxide and pulls an oxygen away from that, so that oxygen gas is formed, 2 molecules of water, and the newly reformed iodide ion. You can order potassium iodide from any chemical supplier, but it's not something that most people have lying around their house. So the other method is to use yeast. Yeast is a tiny living organism, and like most organisms, it produces an enzyme called catalase. The function of catalase is to break down hydrogen peroxide, because hydrogen peroxide on its own can be very damaging to cell and cellular components. The exact way that catalase works is beyond the scope of this demonstration, but you can see the effects of it right away. The foam produced is caused by the rapid decomposition of hydrogen peroxide into water and oxygen. The oxygen forms bubbles, and these bubbles are trapped by the dish soap. If you did this without the dish soap, you would still see bubbles, but you wouldn't get an impressive foam. In fact, this is a common test done in microbiology labs, and it's used to identify organisms. Organisms can be classified into 2 groups, catalase positive and catalase negative, meaning they produce catalase or they don't. The majority of bacteria are catalase positive, and this includes things like Staphylococcus, Salmonella, and the bacteria that cause body odor, tuberculosis, and the plague. Don't worry though. Most catalase-positive organisms are not pathogenic. There are tons of bacteria species on this planet, and only some of them cause human disease. Important catalase-negative bacteria include Streptococcus and Clostridium, which is the genus that includes the bacteria that causes tetanus. To help distinguish between these organisms, especially things like Streptococcus and Staphylococcus, which can look similar under a microscope, the researcher or lab technician will take a small sample of the bacteria culture and expose it to a bit of hydrogen peroxide. If it forms bubbles, that indicates the presence of catalase, and that organism is said to be catalase positive. That's the same reaction that we see here, minus the dish soap. If it does not produce any bubbles, it is said to be catalase negative, and that helps the researcher identify what kind of organism they're dealing with. Your body produces small amounts of hydrogen peroxide in multiple ways. One of these ways is by the immune system, and it's to kill invading bacteria. In some people, the enzyme that's responsible for producing hydrogen peroxide is defective, and they're unable to produce much hydrogen peroxide. When an organism that is catalase positive infects an individual that has this disease, they will be unable to fight off the infection, and it can become a chronic infection. This is called chronic granulomatous disease, and that's because the immune system, when it can't destroy the bacteria using the hydrogen peroxide system, it will instead wall up those organisms in a type of pathogen prison surrounded by macrophages to keep that pathogen sealed away and prevent it from causing harm. This pathogen prison is called a granuloma, and unfortunately, it can sometimes cause problems of its own. Dilute hydrogen peroxide is used for disinfecting cuts, and stronger solutions are used for disinfecting tools and sometimes even entire rooms. High concentration hydrogen peroxide is also used as a propellant in rocketry. Remember how I said concentrated hydrogen peroxide was dangerous to handle? That's because in addition to being corrosive, it is also potentially explosive, and it has to be stored in very specific conditions, including in containers that are of the right material and shape. Even the 30% concentrations that you find at beauty supply stores for bleaching hair can be potentially dangerous. At concentrations of 70% or greater, they can become very explosive. Concentrations of around 90% are used as propellants in rocketry. They are effective propellants because they start a chain reaction. In addition to decomposing into water and oxygen, this reaction also produces heat. It's called exothermic. Heat is also a trigger for the decomposition. So what happens in a hydrogen peroxide fueled rocket is that part of that solution will be thermally decomposed, and the heat that is produced from that decomposition reaction will go on to trigger further decomposition in the rest of the solution. The water that is produced by this reaction will be turned to steam, and the steam exiting the rocket is what gives it its propulsion. But even lower concentration of hydrogen peroxide, like the 3% solution that I'm using, produce heat. This reaction will get warm, but not dangerously hot. If this reaction is done with higher concentrations of hydrogen peroxide, it has the potential to get very hot. It is generally safe to handle drugstore concentration levels of hydrogen peroxide, but just to be safe, I'm wearing my safety goggles and gloves. Even at this dilute strength, it can be irritating to skin and eyes, and the number one rule of lab safety is better safe than sorry. It's good practice to always wear the appropriate protective equipment for whatever you're doing, and to handle chemicals with respect. The foam itself may still contain unreacted hydrogen peroxide, which is why I don't want to touch it. If you use higher concentrations of hydrogen peroxide, it can be dangerous to touch the foam, as the unreacted hydrogen peroxide may be corrosive. So as you can see, This reaction, in addition to looking really cool, demonstrates a lot of important principles that are used in everything from microbiology and medicine to physics and rocketry,